Hello, my name is Hannah Petry, and today I will be presenting my poster presentation about the effects of tart cherry on the lifespan of Alzheimer's C. elegans. So first, a little bit of background about our project. We are mainly focusing on Alzheimer's disease. Alzheimer's disease is the decrease in memory of these patients, which is characterized by the presence of amyloid plaques and neurofibrillary tangles, which is caused by an increase of neuroinflammation and oxidative stress. What we're looking at to help treat Alzheimer's disease are tart cherries. We chose tart cherries because they are rich in antioxidants and anti-inflammatories, thanks to having properties of being those anthocyanins. A model that we're using for our research are called C. elegans. C. elegans are a great model for our lifespan research because they are free-living, non-parasitic nematodes, which have about 65% of their genes are homologous to humans, as well as their short lifespan of 3 weeks, which is very beneficial when replicating data for lifespan research. The specific strain that we're using is called GMC 101 C. elegans, and they are similar to human Alzheimer's disease because they also accumulate high amounts of those amyloid plaques that we discussed earlier. And also, previous research in our lab, when we treated wild type C. elegans with tart cherries, we found that it significantly increased lifespan. As we can look at our data, our control group who had no tart cherry had a mean lifespan of 13.3 days, followed by 14.9 days for our 3 microgram per milliliter group, 16.8 days for our 6 microgram per milliliter group, and 16.7 days for our 12 microgram per milliliter group, with our 6 and 12 microgram per milliliter group being the two that significantly increase this lifespan. So our hypothesis for this study is that tart cherry extracts are going to increase the lifespan in Alzheimer's disease C. elegans. So the impact of this project and how we can help benefit society is that we're hoping that we're going to help increase the life expectancy of patients who do have Alzheimer's disease through more safe dietary interventions. So how we conducted this project is that we laid adult C. elegans on Petri plates for about 4 hours. We came back and removed these adults and allowed the eggs that they laid to grow for about 72 hours. And then we transferred these now age-synchronized adults to microfluidic devices where they are going to live for the rest of their life. We had 3 treatment groups, with our first group being a control group being fed normal 20 milligrams per milliliter of E. coli, which is their basic food they usually have. We then had our low dose group who had 6 micrograms per milliliter of tart cherry on top of having that 20 milligrams per milliliter of E. coli. And our high dose group had 12 micrograms per milliliter of tart cherry on top of that E. coli food as well. We then used this NEMA Life machine to record a 90-second video daily of our C. elegans to determine how many are alive each day. So what we found from this study is that the treatment of tart cherry significantly increased the lifespan in our 12 microgram per milliliter group compared to our control. Our control group had a median lifespan of 13 days, followed by 15 days for our 6 microgram per milliliter group, and 15.5 days for our 12 microgram per milliliter group. So conclusion from this, we found that 12 micrograms per milliliter of tart cherry significantly increased Alzheimer's disease C. elegans lifespan. And when converting this to humans, this will be about 12 to 14 tart cherries per day. And in future studies, we're hoping to look for more of the effects of tart cherry on different strains of Alzheimer's disease C. elegans, as well as the effects of tart cherry on different Alzheimer's disease-related genes, as well as the effect of tart cherry components such as anthocyanin on Alzheimer's. Here are some of our references that we used when looking to our study and researching different benefits. And we'd also like to thank the Nutrigenomics, Inflammation, and Obesity Research Lab and everyone in there for their help and guidance for this project, as well as the funding from the USDA and College of Human Sciences. Thank you.